السلام عليكم جماعات. Agrochemists, peptides and proteins و job purification, separation, and checking. Uh, uh, as we said, proteins and peptides are polymers of amino acids uh, or chains of amino acid. Two amino acid molecule can be covalently joined through a substitute amide linkage turn a peptide bond to yield a dipeptide. So in carbohydrate, in polysaccharide, the monomer were monosaccharides and they were joined together through glycosidic bond. Here we have amino acid as a monomers and they are joined together through a peptide bond. It's an amide linkage. Later we'll see how it's formed and in detail. Such a linkage is formed by removal of the elements of water. So formation of peptide bond is a dehydration reaction, removing of water molecule, hydrogen from one amino acid and hydroxyl group from the other amino acid. So this will leave water from the alpha carboxyl group of one amino acid and the alpha amino group of another. Peptide bond formation is an example of condensation reaction. So condensation reaction is basically removing water molecule or formation of water molecule, a common class of reactions in living cells. Under standard biochemical condition, the equilibrium for the reaction favor the amino acid over the dipeptide. To make the reaction thermodynamically more favorable, the carboxyl group must be chemically modified or activated so that the hydroxyl group can be more readily eliminated. Three amino acids can be joined by two peptide bonds to form a tripeptide. Similarly, amino acid can be linked to form tetrapeptide, pentapeptide, and so forth. When a few amino acids are joined in this fashion, the structure is called an oligopeptide. When many amino acids are joined, the product is called a polypeptide. So if we have few amino acids in the chain, up to 20 amino acids, this molecule called oligopeptide. If we have more, like hundreds and thousands, that molecule would be called polypeptide. In a peptide, the amino acid residue at the end with a free amino acid group is the amino terminal residue. At the other end, which has a free hydroxyl group, free a carboxyl group, is the carboxy terminal residue. So you know, in any amino acid we have amino group and we have carboxyl group. So so the chain, so long chain of oligopeptide or polypeptide so here if we have this amino acid, at what end we will have amino group. All these are amino acids, many amino acid, amino, and here we have carboxyl group. That's what I mean. Although hydrolysis of a peptide bond is an exogenic reaction, it occurs slowly because of its high activation energy. Activation energy is the energy needed to break down a bond. So hydrolysis of dipeptide or tripeptide is exogenic, give energy, uh, but it's it at the beginning it needs energy, then it will release energy. It needs energy at the beginning because of its high activation energy. As a result, the peptide bonding proteins are quite stable because of its high activation energy, with an average half-life of about 7 years under most intracellular conditions. This is amino acid. You see here, it's alpha carbon, carboxyl group, and this is uh, amino group. This is another amino acid. Uh, the hydrogen from the amino group will be removed, and hydroxyl group from the carboxyl group will be removed. Uh, this is a dipeptide, and this is the amide linkage or peptide bond, you can name it. Many small peptides exert their effect at very low concentration. For example, a number of vertebrate hormones are small peptides. These include oxytocin. So we have some peptides uh, have 3 amino acid, 4 amino acid, but they have a huge impact. They carry out important physiological functions like oxytocin. They only have 9 amino acid residue, which is secreted by the posterior pituitary and stimulate uterine contraction. Uh, Bradykinin, 9 residues, which inhibit inflammation of tissues. And thyrotropin releasing factor, only 3 residues of amino acid is formed in the hypothalamus and stimulate the release of another hormone, thyrotropin, from the interior pituitary gland. Some extremely toxic mushroom poisons such as amanitin are also small peptides, and as are many antibiotics. Glucagon, another pancreatic hormone, has 29 residues, and it opposes the action of insulin, which is also a peptide. Many hormones are basically peptides or oligopeptide or peptide. We will discuss hormones later in detail. Working with the proteins. So, what are the methods that we can use to purify and separate proteins? A pure preparation is essential before a protein's properties and activities can be determined. So sometimes some proteins can act as a drug, like insulin. Uh, Sometimes we need to know the functions of protein, activity of them. So to do this, to carry out this, we need to separate them, purify them at the beginning. Given that cells contain thousands of different kinds of protein, how can one protein be purified? Methods for separating proteins take advantage of properties that vary from one protein to the next, including size, charge, and binding protein. Uh, Obviously, one protein is different from all other proteins. They differ in size, in charge, in affinity, in the structure, in so many things. They differ from each other. So the technique that separate protein take advantage of these properties or these features. The first step in any protein purification procedure is to break down these cells, releasing their proteins into a solution called crude extract. So, proteins are present inside the cell. So, at the beginning, we need to break down the cell, uh, destroy the cell to a solution called crude extract. Then, if necessary, differential centrifugation can be used to prepare subcellular fraction or to isolate subspecific organelles. Cell contain many organelles. Sometimes we need different centrifuge this crude extract to separate organelles from each other. Heavy suborganelles will goes down, and light organelles will go up. Once the extract or organelles preparation is ready, various methods are available for purifying one or more of the protein it contains. Commonly, the extract is subjected to treatment that separate the protein into different fractions based on the properties such as size, charge, process referred to as fractionation. The most powerful method for fractionating protein make use of column chromatography. This is a potent technique to separate protein which take advantage of different in protein charge, size, binding affinity and all other proteins. This is column chromatography. This is the beginning. We will put crude extract here, the protein solution here, and will slowly come down to the column. Here, after some time, we'll open the burette. After some time, you see here lots of bands. At the end, we will have each and every single band of this is a specific protein. So here this color is one protein, we can separate them. Then once it reach this color, we will remove that beaker and bring another one to let this color goes into it. Once it finish, we can use another beaker. So this way we will have many beakers with different proteins. That's how column chromatography works. A porous solid material with appropriate chemical property, the stationary phase, the phase that's inside the column, held in a column, and buffer solution, the mobile phase, the percolate through it. The protein containing solution laid on the top of the column percolates through the solid matrix as an ever expanding mass. So gradually this band will expand to make the separation easier. Individual proteins migrate faster or more slowly through the column depending on their properties. So here like small protein will migrate faster, bigger will migrate slower, etc. Individual proteins faster. For example, in cation exchange chromatography, this is another technique to separate the protein. The solid matrix or the stationary phase has a negatively charged, whereas proteins in the cation exchange chromatography, the solid matrix has a negatively charged group. So in the mobile phase, protein with a net positive charge will migrate more slowly because it will be attracted or retarded by the stationary phase. That's why they migrate slower. But protein with a negative charge will migrate faster because there's no any attraction or retardation by the stationary phase. As the length of the column increases, the resolution of two types of protein with different net charges improve. So as the column is longer, the separation and resolution of the separation will be better. We have another technique called size exclusion chromatography. Separate protein according to size. In this method, large protein emerge from the column sooner than small ones, somewhat counterintuitive result. So here, larger proteins migrate. Be careful here. Larger protein migrate faster, whereas a small protein migrate slower. Why? we will give you the answer very soon. The solid phase consists, this is why. The solid phase consists of beads. نفقه لنا of solid phase. Konishken. Veja. Evet mazan نوچن وان konishka da. Kon وقت نفقه درجو أو نوچن تدا. هما راست التنخاري. بس أبد بجيك تشرفان كنادا تشتو حتى بقى تبقى وكنا جبركي رخ كي تشتو تيتن لو ما تأخير البتن أبي سبب مولكيولات بجيك تأخير مثلا زيكاتين خارج. Large proteins cannot enter the cavities and so take a short and rapid path through the column around the big small protein enter the cavities and migrate through the column more slowly as a result. We have another technique called affinity chromatography. It's based on the binding affinity of proteins, the beads. So here it depends on the affinity between the protein and the stationary phase. As the affinity in some protein will have a more affinity to the chromatography, so to the stationary phase, so they will migrate slower, and some of them will not have any affinity. That's why they will migrate faster. Protein with affinity for this particular group will bind to the beads in the chromatography migration will be retarded as a result. The most famous technique to separate a protein is electrophoresis. This takes advantage that protein can be visualized as well as separated, permitting a researcher to estimate quickly the number of different proteins in a mixture or the degree of purity of proteins preparation. Also, electrophoresis allows determination of crucial properties of proteins such as its isoelectric point. Through this technique, we can separate the protein, we can measure isoelectric point, we can measure molecular weight, and some other property. Electrophoresis of protein is generally carried out in gels made up of the cross-linked polymer polyacrylamide. So here the stationary phase is a gel. It's a gel cross-linked polymer polyacrylamide gel. Polyacrylamide gels act as a molecular sieve عائقة بروتيني, slowing the migration of protein approximately in proportion to their charge to mass ratio. This is gel electrophoresis. This is the gel here. All these, the yellow, the blue color is gels. This is the wells. Here the place where the protein can be put in. Then this is the negative pole. This is the positive pole. So protein with negatively charged will migrate faster to the positive charge, whereas as the positive charge decreases, the migration will be slower until all the protein get separated. At the end we will have this band here. So each and single band is different protein here. And that's it.